Dr. Balmes is a professor of medicine at the UC San Francisco. He's chief of the Division of Occupational and Environmental Medicine at San Francisco General Hospital. He also attends as an attending physician there, so not only does he do research and teaching, but he also actually sees patients. In addition, he's director of the Human Exposure Laboratory at the Lung Biology Center there, and he's a professor of environmental health sciences in the School of Public Health right across the bay over here in UC Berkeley. So, and finally, he was appointed in December '07 by our previous governor, Schwarzenegger, to be the physician member of the California Air Resources Board. So we're happy about that because he actually knows a lot of the science. So, yeah. And Dr. Balmes has been conducting air pollution health effects research for many, many years now. So welcome, John. Thank you.
Thanks, Melanie. You might be happy that I'm on the Air Resources Board, but I don't think the trucking industry is particularly happy. They have called me an environmental Nazi and various other cool names, because they don't believe the science with regard to diesel and health effects. So I'm here representing the UC Berkeley-Stanford Children's Environmental Health Center, which is not a full PO1 Children's Environmental Health Center like Brenda's and Asa's center at UC Berkeley or Elaine's at UW. We have a formative center grant. It's only for 3 years, and we're anxiously awaiting an RFA coming out hopefully next month with regard to a chance to build for the future and become a full PO1. And we've been doing air pollution health effects research, our group at UC Berkeley, for over 12 years, mostly centered in Fresno in the Central Valley, California. And we parlayed that work into a successful grant for one of these formative centers. And the Fresno study that this was built on was called FACES, Fresno Asthmatic Children's Environment Study. And now we have CHAPS, Children's Health and Air Pollution Study, San Joaquin Valley. And Ira Tager is our principal investigator. So why the San Joaquin Valley? Probably since most of you are from California, this is self-explanatory, but for those who aren't, it's got very high air pollution, primarily mobile source. And then there's the valley topography. So it's especially to the east are the Sierra, and the Sierra basically traps the air pollution that is partially sourced from mobile sources in the Central Valley, but also San Francisco's bad air actually blows down into the Fresno area. It's rapidly growing in terms of population. It's a very diverse population ethnically, and unfortunately, it's really been hit hard by the recession. Unemployment in Fresno is like 20%, or at least the smaller towns around Fresno. So this is the Lung Association State of the Air report for last year, and you can see that Bakersfield, which is in the south end of the valley, ranked #1 for most polluted city in terms of particulate pollution, and Fresno's #2. And there's various other cities of the Central Valley as well in there. So before I launch into a discussion of some of the work we've been doing, the actual research, just to give you a sense of how we're structured, there's an administrative core, a community outreach and translation core, and then we have 3 projects, and the exposure is all air pollution of various types. There's a birth weight and prematurity project, a birth defects project, and an asthma and regulatory T cells project, which is what I'm going to talk about today. And we have a biostatistics core and exposure assessment core. And our exposure assessment core is focused on measuring ambient air pollutants. And just instead of having an acknowledgement slide at the end, have the key personnel here, and I won't go through all these names, but you may recognize a a few folks. And it's kind of a little bit interesting that they asked me to be head of the Community Outreach and Translation Corps. It's something I don't have expertise on, but some of our team are less popular in Fresno than me, so I won the popularity contest in terms of the local community groups. And I might say that those community groups actually nominated me to be on the Air Resources Board, so the feeling is mutual. So the first project is on birth weight and prematurity, and the basic hypothesis was that adverse pregnancy outcomes related to birth weight and prematurity are increased with exposure to ambient particulate matter and bioaerosol. In particular, we've been interested in endotoxin in terms of a component of bioaerosol. And bottom line is we have high-quality exposure data that we've been collecting in the Fresno area for, again, over a decade, and then birth records from 4 of the Central Valley counties. And because the birth records have the residence of the mother at the time of birth, we can add various GIS contextual factors, and that's one of the things what we're doing in particular. Then project 2 is for birth defects. Gary Shaw at Stanford is the leader of this project, and he's involved in project 1 as well. And the hypothesis is exposure to specific air pollutants during organogenesis is associated with specific birth defects. And here, in order to take advantage of the fact that Bakersfield has higher polycyclic aromatic hydrocarbon levels than Fresno, we expanded our monitoring to Bakersfield and also expanded our network of endotoxin monitoring. And so here the basic point is we have high quality exposure data, and we use California Birth Defects Registry data and look at associations. So project 3, which I'm going to talk about in more detail, and is headed by Kari Nadeau at Stanford, who is a pediatric allergist, basic immunologist, who came to see Ira, Tager, and me a number of years ago because she was sort of frustrated she couldn't find anybody at Stanford who would want to collaborate with her in terms of environmental exposures and asthma. And we were really excited because she's a very talented basic scientist and a good clinician. And when Amy Kyle asked me who from our center should give a talk today about this topic, I said, well, it's Kari's work, she should do it. And then Amy said, is she going to talk with a lot of fancy molecular biological technique type of jargon? And I said, probably. And she said, okay, John, you have to translate. So I'm a co-author of the work that is being presented, but if you press me too hard on specific molecular biological data, I might be limited. So anyway, Kari's hypothesis is that T regulatory cells are dysfunctional in children exposed to high levels of ambient air pollution, and this contributes to the burden of asthma. Now, what are T regulatory cells? I'll show you in a slide or two, but they are sort of the controller cells of the immune response, especially the cell-mediated immune response. And so T helper and T suppressor cells, so-called T effector cells, are controlled by the T regulatory cells. And as I mentioned, Kari's lab uses state-of-the-art molecular biological techniques to understand mechanisms by which air pollutants affect asthma. So why are we interested in asthma? Well, I probably don't have to convince this audience this is an important disease in children. And it's sort of a classic gene-environment interaction disease. There's clearly genetic risk factors for asthma. Asthma runs in families, and allergies run in families, and people who are allergic are at higher risk for developing asthma, but plenty of allergic people don't ever develop asthma. So there has to be other hits, and some of those hits might be air pollutant, though others might be viral infections. And of course, you can get a viral infection and be exposed to air pollution at the same time, which may be a double whammy. So there's a lot of healthcare dollars being spent to deal with the asthma epidemic. which is really a disease of the developed world. I think most of you know that, but this has become strikingly apparent to me since I've been doing work in Guatemala for the last 7 years. And the PI of the indoor air pollution from biomass smoke study that I'm involved with in Guatemala, Kirk Smith, my colleague at Berkeley, came to me and said, you know, I've got this cohort down in Guatemala exposed to biomass smoke. I want you to help me study biomass smoke and asthma. And in this setting, it's the highlands of western Guatemala. And I thought about it and I said, Kirk, there's not any asthma there. We won't be able to study that. So we're studying the effect of biomass smoke on the growth of lung function in these kids. Because, again, I've been down there for 7 years and we rarely diagnose a kid with asthma, though we try hard. So, again, I think most of you know that, but asthma is definitely a disease associated with the Western lifestyle. And one of the components of the Western lifestyle is exposure to ambient air pollution of a traffic source. And that's really the focus of the air pollution work in our center so far. So here's T regulatory cells, or Tregs. They control or suppress the function of other immune cells. And Foxp3 is a transcription factor that's associated with T regulatory function, and it's been a focus of Dr. Nadeau's work. And it's already been documented by others that children lacking Foxp3 have severe allergies, asthma, GI disease, and at risk for type 1 diabetes. And T regulatory cells can inhibit effector T cells, and those are either T helper or T suppressor cells. And then the T helper cells are split to Th1 and Th2. I think many of you know about this paradigm. And it's Th2 cells that are increased in, and their activity is increased in individuals with allergic disease such as asthma. And the T regulatory cells can dampen down this effector cell response, and so can dampen down allergies. So Kari has been interested in what environmental factors can decrease Treg function in asthma, and she hypothesized that these environmental factors might work through Foxp3 expression. So when Kari first came to us, before we had the P20 grant, we had the Fresno Asthmatic Children's Environment Study data, and we had 315 children, 8 to 12 years of age, enrolled in the study. At that point, when she came to us, we had 9 years of exposure data. We now have 12. And we recruited a sub-cohort of these FACES kids, 71. We had serial spirometry, lung function, and clinical symptom data. And what Kathy Hammond, head of our exposure core, at that time our exposure component of FACES had, was an interest in polycyclic aromatic hydrocarbons, in addition to all the standard criteria pollutants. And she and her grad student, Betsy Noth, had come up with, they basically mapped Fresno with regard to polyaromatic hydrocarbon levels. We had multiple samples taken at homes of our participants scattered throughout the city, and then we had a central site monitoring system co-located with the EPA CARB monitoring station. For those of you who know Fresno, First Street. And then we had several neighborhood monitoring stations that we set up at schools. So we had an extensive monitoring system for polycyclic aromatic hydrocarbons. I think I'll say PAHs, it's easier. And we were able to create a map, a surface, so that we could estimate exposures anywhere in the city. I'll show you that map, I think, on the next slide. And then we came up with a study design to compare the Fresno asthmatic children from FACES with control kids, age and sex matched, without asthma or allergies. We had 40 of those. And then Kari recruited 30 kids from her practice at Stanford, also age matched and sex matched to the children with asthma, 30 of those. And then we came up with some Stanford controls that were age matched and sex matched with the kids from Fresno who didn't have asthma or allergies. And the Stanford group had to be over 6 because our kids had started, we recruited kids at FACES at age 6. And they had to live in Palo Alto for 6 years at a specific residence and less than or greater than 30 meters from a major highway. Here's a bit of our mapping of PAH cumulative exposure over 12 months. And so this actually isn't showing the surface as much as showing relative levels of exposure throughout the city. And you can see exposure tends to be highest along the freeway corridors where there's the most vehicular traffic, especially truck and bus traffic. And so Kari was able to look at regulatory T cells by molecular techniques, including epigenetic data, which I'll share with you, as well as effector T cell data. I'm not going to really show much of the effector T cell data. She looked at interactions between the 2 cells. Again, I'm not going to show that. And then we had developed an asthma severity score using the Global Initiative for Asthma sort of international scheme. We'd adapted that for FACES, and we used that for Kari's Stanford kids as well. And we had lung function on the FACES kids, and we obtained it on Kari's Stanford subjects. And this is, to me, was pretty surprising data when I first saw it. I mean, it was almost so good that I didn't believe it. This has been published, and it's available in the Journal of Allergy and Clinical Immunology. And so FA is the Fresno asthmatic kids, FNA, Fresno non-asthmatic kids, SA, Stanford asthma kids, and SNA, Stanford non-asthmatic kids. And this is percent of Treg cells that express Foxp3. And again, expressing Foxp3 is good. And you can see that there's a nice sort of step function. What was surprising to me was that the Fresno non-asthmatic kids actually had, almost, they were similar to the Stanford asthmatic kids in terms of the level of Foxp3 expression. And definitely the Fresno asthmatic kids were clearly lower. Then in the panel B, it's expression of Foxp3 in the Treg cells, and then actual functional assay of the T regulatory cells, see how well they worked. And again, a pretty impressive linear relationship between the expression of Foxp3 and how well the cells worked. And then we looked at Foxp3 expression in the Treg cells versus asthma symptom score, and there was, again, a nice effect here. And then this is lung function. And Treg function was directly associated with predicted lung function in the Fresno asthmatic subjects. So this got us, pretty excited. This is more work showing that there was a downstream effect of the T regulatory cells, that there was Th2 skewing in the Fresno asthmatic kids. They had less Foxp3 expression in T regulatory cells. So the T regulatory cells were less effective at inhibiting Th2 expression of cytokines is what we show here. IL-4, interleukin-4, and interleukin-13 are two markers of Th2 cell activity. And the Fresno asthmatic kids had, high levels, higher than the Stanford asthmatic kids. And then the Fresno non-asthmatic kids had higher levels than the Stanford non-asthmatic kids. So that got us to think, what's different about Fresno air than Palo Alto air? A lot of things, but PAHs were compounds of interest to us, and PAHs in Fresno are about 7 times higher in Fresno than in Palo Alto. Actually, we don't have measurements from Palo Alto. We used the EPA CARB site in Redwood City, which is close to Palo Alto, not exactly the same. And this is just comparing the levels. So this is a central site in Fresno versus the Redwood monitoring station, and about 7 times higher for total PAHs. There also were differences in terms of the fine PM and ozone as well. So we're not trying to claim that PAHs are the whole story here, but we thought they might be part of the story. And we looked in our entire phases cohort, which was 315 total, but we only had pH exposure estimates that we felt confident about for 283. And in that cohort, we looked at estimated pH exposure and wheezing, and odds ratios for asthmatic kids exposed to PAHs and wheeze ranged from 1.01 to 1.10. Statistically significant, not super strong, but statistically significant. And when we looked at one pH in particular, Phenanthrene, which was mentioned earlier today, is a semi-volatile phenanthrene, pH, and one that's known to affect immune function in the laboratory. Phenanthrene was found to have a higher relative impact on wheeze than the other pHs. And this is coming out in a paper that's in press in the Journal of Exposure Science and Environmental Epidemiology. So we had evidence that Fresno asthmatic kids had abnormal immune function skewed towards the allergic phenotype, and we had evidence that the PAHs were associated with wheeze among asthmatic kids in Fresno. So Kari hypothesized that PAHs might increase methylation of Foxp3 and therefore decrease Treg function. So this is epigenetic work. I think most of you are familiar with epigenetics in general. So this is methylation changes the expression of the gene. And Foxp3 has multiple sites with CPG regions that can be methylated. These are both promoter sites and intronic sites. And lo and behold, there was a nice step function, if you will, again with the Fresno asthmatic kids having the most methylated CPG in Foxp3, which means that Foxp3 is going to be, its expression is going to be downregulated. And so the effector T cells will be more active. And we went on to look at pH exposure from our exposure map and degree of methylation of CPG islands, and there was a very strong correlation. And then we also looked at pH exposure and Treg function, and that was also strongly related. So Kari has gone on to do some further laboratory work. It's unpublished, but she thinks it's cool enough that she's going to submit it to Nature Medicine as soon as we have the knockout mice work to confirm the ex vivo cellular work here. But she's shown that phenanthrene increases CPG island methylation in the laboratory. in the Foxp3 locus. And because she was trying to submit this to a really good journal, she's made sure that she showed that it wasn't just methylation of the gene, but it also affected gene expression and then protein expression, this is methylation of the gene. You can see that the diluent had no effect, and phenanthrene increased methylation. And then this is expression of RNA for Foxp3. Diluent had no effect. There was a big effect of phenanthrene, and then this is protein expression in the cells, also showing an effect. So, in summary, we've shown clinical outcomes, really severity of asthma, both by symptoms and lung function, are inversely correlated with the presence of memory Treg cells with stable Foxp3 expression. That was pretty new and exciting. Foxp3 is unstable in Treg cells after methylation of promoter and intronic regions. Methylation of Foxp3 is associated with PAH exposure in our Fresno population. PAH exposure is associated with Wheeze in asthmatic children in Fresno, in a larger cohort. And then she's gone on to show ex vivo studies using phenanthrene in culture with Treg cells have shown that this pH increases methylation of Foxp3 loci. And what she's actively working on is mechanisms by which phenanthrene and other pHs may induce or exacerbate asthma. And I hesitate to close with this slide. This is Kari's, so don't blame me. So this is her basic model, which she brought to us, and we're glad that she did. So environmental air pollutants, in this case, we're talking about pH, cause methylation of promoter region of Foxp3. She feels, based on data that would be in her Nature Medicine paper, that this is through the aryl hydrocarbon receptor, which binds pHs. And, whoops, I didn't want to do that. And so when you have decreased Foxp3 expression, you're going to have decreased suppression of effector T cell function that promotes allergy. And it might also affect the airway inflammatory response to exposure to allergens as well. And so we're trying to fill in the various steps in the pathway by which air pollutants in general, but pHs in specific, might be affecting allergy and asthma. And with that, I thank you for your attention.